Oxidative phosphorylation is one of the pathways used to make ATP. In eukaryotes, oxidative phosphorylation occurs in the mitochondria. A chain of protein complexes 1 through 4 pump protons into the inner membrane space. The protons flow back into the matrix through ATP synthase, which generates ATP. ATP synthase, also known as complex 5, is the last enzyme in the oxidative phosphorylation pathway.
The ATP synthase enzyme is one of the key regulatory enzymes that can reversibly produce or hydrolyze ATP. ATP is an energy storage molecule that is most commonly referred to as the energy currency of the cell because it allows the body to perform all of its vital chemical reactions. ATP is formed by the substrates adenosine diphosphate and inorganic phosphate. ATP synthase is a complex structure consisting of two major domains, F0 and F1. The F1 domain is the molecular machine that catalyzes the phosphorylation of ADP to ATP, utilizing rotational energy. It is a spherical structure that sticks out into the mitochondrial matrix and is anchored to the membrane by a stator to prevent rotation. It consists of 6 nucleotide binding sites, which are 3 non-catalytic alpha subunits and 3 catalytic beta subunits. The F0 domain is the proton turbine that is motorized by the translocation of protons down a proton concentration gradient that exists across the mitochondrial membrane due to the ETC during respiration. It is a cylindrical structure capable of rotation when driven by translocated protons. It is also linked to the central stalk that can revolve inside the F1 domain to cause catalytic conformational changes in each nucleotide binding site.
The mechanism of ATP synthesis within the F1 subunit involves the active site of beta subunits cycling between 3 different states. At the open state, ADP and phosphate enter the active site. The enzyme closes around the substrate loosely in the loose state. Then the enzyme undergoes a change in shape, forcing the substrates together, generating ATP, resulting in a tight state. Finally, the active site returns back to the open state, releasing ATP in the process, and is then ready for the next cycle of ATP production.
As previously mentioned, ATP synthesis by the synthase is coupled to the flow of protons across the inner mitochondrial membrane. Certain genetic defects in the synthase itself can cause dysfunction of its parts and possible loss of coupling. The loss of this coupling results in the flow of protons through the synthase without the generation of ATP. This leads to the subsequent loss of the proton concentration gradient across the membrane and thus the loss of the energy needed to power the synthesis of ATP.
Uncoupling of these two processes has been shown in several cellular studies to result in the loss of mitochondrial DNA. This explains the accumulation of mitochondrial genetic defects over time in individuals with ATP synthase deficiency. These studies also show the importance of the synthase in maintaining the integrity of functional mitochondria and its DNA.